let's jump right into this explainer because today we're going on an evidence quest. Have you ever stopped and wondered why exactly your eye doctor prescribes a very specific drop, or maybe why they push a particular vitamin? Well, it's definitely not guesswork. It is bedrock science. And here's our roadmap for today. We're starting at the front of the eye with herpetic disease, working our way to the back with macular degeneration, and finally wrapping up with drug toxicity screening. Let's get into it. So, how do doctors actually know what works for your eyes? They rely on massive, rigorous clinical trials. These are the studies that test our theories, track the real-world results, and honestly, sometimes they completely overturn everything we thought we knew about practicing medicine. Which brings us to our first stop, herpetic eye disease and establishing targeted antiviral dosing. This all goes back to the legendary herpetic eye disease study, or HEADS for short. This study completely changed the game by making treatments way kinder to the patient's eye. For instance, we learned that ganciclovir 0.15% gel is significantly less toxic to the ocular surface than those older trifluoridine drops. Plus, because it's a gel, you don't have to use it nearly as often. But maybe the biggest takeaway here? Oral antivirals work really well. In fact, they might actually be your go-to depending on the patient. Like, say if they already have severe surface disease or just can't handle putting drops in their eyes. And we can see exactly how these findings translate to different types of keratitis. Look at stromal keratitis. The main play there is topical or oral antivirals, but you absolutely need that secondary addition of topical corticosteroids with a really long taper. On the flip side, if you're dealing with endothelial keratitis, you're relying heavily on oral antivirals right out of the gate, paired immediately with prednisolone acetate drops that you carefully taper over 10 weeks. It's not a one-size-fits-all thing. It's a completely tailored approach based directly on that trial data. Okay, moving right along to part 2, ARDS 1 and tackling the posterior segment. Let's shift to the back of the eye and talk about the absolute heavyweight of vision loss, age-related macular degeneration, or AMD. Just as a quick refresher, this is an acquired retinal disorder where we see physical changes in the fundus. We're talking pigmentary atrophy, drusen, lipofuscin deposits, and that exudative elevation of the outer retinal complex. Now, when the ARDS 1 trial drops, it gave us one phenomenal number, 25%. Researchers tested a very specific mix of antioxidants and zinc on over 4,000 people. And what they found was that for patients with high-risk AMD, this exact formulation led to a massive 25% decreased risk of progressing to advanced AMD, plus a 19% drop in the risk of vision loss. Honestly, that is a huge victory for preventative eye care. But, and there's a big but here, ARDS 1 came with a pretty terrifying clinical plot twist. The original formula used 15 milligrams of beta-carotene. Well, researchers quickly realized that beta-carotene is actually contraindicated for smokers because it actively increases their risk of lung cancer. I mean, obviously, you can't hand a patient a pill to save their eyesight if it's literally boosting their chances of getting cancer. So, that glaring issue leads us right into Chapter 3, refining those formulas with the sequel study RADS2. They had to fix the beta-carotene problem, so the ARDS2 trial tested a new idea. What if we just take out that risky beta-carotene completely and swap in dietary lutein and zeaxanthin instead? Oh, and they also threw in some omega-3 fatty acids, DHA and EPA, just to see if they could keep that incredible protective effect without, putting smokers in danger. So what happened? Well, the results were actually pretty fascinating. Adding all those extra ingredients didn't magically reduce advanced AMD progression any more than the original formula did overall. However, it did exactly what it needed to do for former smokers. Lutein and zeaxanthin turned out to be a completely safe, appropriate substitute. Plus, as a little bonus discovery, patients who didn't get much lutein and zeaxanthin in their normal, everyday diets actually saw a much bigger protective boost from the supplements. Problem solved. Keeping the AMD momentum going, let's jump into Section 4, the CATT study, which I like to think of as the ultimate neovascular heavyweight bout. In this corner, we have treating wet AMD with Lucentis. That's the standard, on-label treatment. And in the other corner, we've got Avastin, the off-label underdog. They put these two head-to-head using the exact same playbook. Initial monthly injections, followed by either strict monthly visits or a PRN approach, which just means treating as needed. The goal was simple: figure out which drug actually does the job better. And the punchline of this massive trial? It's a dead tie. After a year of tracking visual acuity, Avastin's efficacy was completely equivalent to Lucentis. They both gave patients fantastic visual gains. But here's the kicker. Avastin does it at a fraction of the cost. I mean, this fundamentally changed the entire economic landscape of treating wet AMD, and it didn't sacrifice a single drop of patient care. A total game changer. All right, let's bring it home with Section 5, screening for retinal toxicity and the AAO guidelines for hydroxychloroquine or HCQ. So, stepping away from treating eye diseases to preventing them, HCQ is a fantastic systemic medication, but it has a dark side. It carries a risk of bilateral bullseye retinopathy that can permanently trash a patient's central vision. To stop that from happening, we have to hawk-eye a few major risk factors. The absolute biggest one, a daily dosage over 5.0 milligrams per kilogram of real weight. But you also have to watch the clock because the risk skyrockets to 20% once a patient hits 20 years of therapy. Throw in concomitant renal disease, which messes with drug clearance, or taking tamoxifen, which literally adds to the toxicity, and you've got a perfect storm to watch out for. Because of all this, we now have a super clear, rigid timeline for screening. Step 1 is getting a baseline exam of the fundus within the very first year of starting the drug. Step 2, if the patient doesn't have any of those extra risk factors we just talked about, you can actually hit the pause button and safely defer annual screening until year 5. But then comes Step 3. Once those annual exams start, you can't just eyeball it. You have to use 10-2 visual fields, or wider ones for Asian patients who tend to show damage further out in the periphery, along with an SDOCT. You're looking for that microscopic thinning of the photoreceptor layers long before any visible damage shows up on the retina. And that brings us to the end of our explainer. But I want to leave you with something to chew on. The trials we mapped out today, HEDS, ARDS, CAT, the HCQ guidelines, they quite literally wrote the playbook for modern eye care. They took out the guesswork and replaced it with hard, undeniable evidence. So, knowing how far we've come, what long-held, totally accepted medical beliefs do you think the next 20 years of clinical trials are going to completely overturn? Keep questioning the science, and thanks for coming on this evidence quest with me today.